So, small group today. Probably the weather kept some of you away, but thanks to everybody who braved the storm out there. I appreciate having someone to interact with. So, before we get started, the Pre-Med Club asked me to show this flyer to everyone. If anyone is interested in a physician-student mentor networking event, there is an event on Wednesday afternoon in the CMFT building. Sounds cool if you're interested in being a doctor. Does anyone from the Pre-Med Club want to say anything? Yeah. All right. Yeah. All right, cool. Yeah, so if you're interested, please sign up, talk to the Pre-Med Club. All right, so the other thing before we get started is I actually want to remind everyone that in 2 weeks your project topics are due. So I just want to spend a couple minutes talking about your projects. Now I can't find the window where I put it. Sounds about right. Oh, it's right there. Okay. So yeah, I just want to briefly go through the project handout. It's posted on Brightspace. It's been there, so hopefully most people have at least taken a look at it. But just to briefly go through it, this project is sort of your chance to do something that is interesting to you about. Yes, I can make this bigger. So yeah, yeah. So the goal of this project is for you to find something that's interesting to you about biochem, and you can pick. Yeah, so I mean it's a research project, right? So you want to use primary sources, secondary sources, sort of look through various types of literature, learning about your topic, and then the format of the project that you presented in is whatever you like. It doesn't have to just be, a research paper. So this is really your chance to hopefully have some fun with something about biochem that's exciting for you. You are allowed to work in pairs if you'd like. You do not have to. You can also work individually. So just keep in mind, if you do work in pairs, there are some parts of this assignment that you do have to do individually, even if you have a partner for the overall project. Hey. So I'm just going through the project because the topic is due in 2 weeks. Yeah. So I won't read through this document in a whole lot of detail, but the format is anything of your choice. There's a few options listed here on this document. As far as content, you're expected to get information from a number of different types of research articles and any other sources that you find helpful, and analyze and synthesize that information into something that you can communicate yourself to demonstrate that you understand. So this is just some of the important dates. So in 2 weeks, that's October 2nd, topic and format selection are due. At that point, let me know if you've chosen to work with a partner. Let me know what topic you picked and some details about it. It doesn't have to be a whole lot of detail, but show me that you put, some amount of thought into your project topic. And if anyone wants to talk to me ahead of time about their project topic, ideas they have, or just to, get a sense of, is this a good place to go? Maybe go somewhere else. Please send me an email, come to office hours, talk to me after class, that whole thing. About a week after that, the paraphrasing practice is due. So you're going to be, you'll do a search for a primary research article that's about your topic. You'll select about a paragraph from that source and paraphrase it in your own words. And then there's a discussion forum on Brightspace where you can post your paraphrase. This is one where even if you're working with a partner, you must do it individually. Pick different paragraphs or different sources to do this with. After that, you'll have a paraphrasing response. So you'll be asked to respond to someone else's paraphrase with some feedback. You can either say, great job, I understand it. You put that in your own words. Or you might comment on something that you might want to change about it. Either this wording is confusing, or this is too similar to the original, or whatever feedback you want to say about it. After that point, you will get a grade of either meets, exceeds, approaches, or fails, depending how you did. And if you need to make a revision, you'll be asked to make a revision about 2 weeks after that. In the beginning of November, you'll have to post a written update of your progress on the topic, or on your project, excuse me. So here's a list of what you'll need to put in that project update. And then finally, around Thanksgiving, if you'd like to submit a rough draft and get feedback, that will be due. The earlier you submit it, the earlier you can get feedback on it. And then finally, at the beginning of December, your final project will be due. And there's more information in the rest of this document talking about things you might consider when you're picking your topic, how you might want to look through literature, how you might go about research, some suggested sources where you might find primary papers. And I won't read this all to you, it's all in the document, but yeah, make sure to maintain academic integrity. Of course, you may not plagiarize. There's some information here about good ways to paraphrase information, some websites you can look through. So please look through this document and get what you need. There's information on the grading breakdown for the project as well in this document. And does anyone have any questions right now? Yeah. Exactly. Yeah, that's the paraphrasing part. Yeah. But even before that, you'll have to submit 2 weeks from today your actual topic. Yeah. But yeah, the paraphrasing part is pick a paragraph or two, paraphrase in your own words, and then give feedback on someone else's paraphrase. And then you will get feedback from me about your paraphrase also. Oh, so everything, there's a forum on Brightspace that should be open. If it's not open already, I'll double check. But everyone should be able to see everyone's paraphrases. So I recommend that you pick, someone's who hasn't already been commented on, so that everyone can get a comment from someone else. But other than that, yeah, you can pick whoever's you like. Any other questions? Cool. Okay. And then one final thing before we get officially started, find my other window again. So I do want to note that last time I got a question about whether you need to memorize the nucleic acid structures, the bases and everything. I don't know. I don't remember exactly how clear I was. I think I said be able to recognize them. I just want to make really clear that I want you to be able to recognize the 5 different bases, know the difference between them. If you see one, be able to tell me what the name of that base is. You don't have to draw it for me, but know what it is. Know if it's a purine or pyrimidine. Also be able to recognize the 2 types of sugars, deoxyribose and ribose. What's the difference between them? I won't ask you to draw them, but know the difference. That's, yeah, in terms of structure. That's, I think, hopefully more clear for everyone. Yes. Yes. Yeah. And then also be able to, say where hydrogen bonds might form with its base pair. Yeah. Yes. Yeah. So you can, instead of memorizing the whole thing, you might want to memorize the differences between them, for example. Yeah. All right. So that is what I wanted to say about that. And then just to go further into that, we're going to start talking about amino acids soon. You will not have to memorize the structures of amino acids. I will always give you a table with all the pictures of all the 20 amino acids. So mostly just really worry about remembering the structures for the nucleic acid bases and sugars and stuff. Okay. So let's continue where we left off last time. So we finished here with the beginning of the question about PCR. And the last question we went over was how many copies of a DNA sequence will be present after 10 PCR cycles. So hopefully everyone's caught up from there. Does anybody have questions about the copies of a DNA sequence from PCR cycles? I know it's been several days and a weekend, and it's not, it's been a minute, so just take a second and think about PCR when we get back into this question. So if there are no more questions on that, I will go on to the next slide. So can anybody tell me how we might use PCR to make a point mutation in a gene? And actually, I'll just give everyone like a couple minutes to think about this, just because, it's been the whole weekend, maybe you're not thinking about it. So if you haven't already thought about this, I'll give you about 3 minutes to think about 5 and 6. All right, so does anybody want to tell me how you can use PCR to make a point mutation in a gene? Yeah. Yes. Yeah. Yeah, exactly. So, so you would use what we call site-directed mutagenesis. Whoops, I forgot to turn my pen on again. so site-directed mutagenesis is just like you described. You use a primer with a mismatch to the gene. So because almost all of the primer does match the template DNA, the primer is still able to anneal, but the primer has, maybe one nucleotide difference. That's enough to make one amino acid change, depending on which nucleotide you change. So as you go on to make copies through PCR, off of that primer, you'll make more and more copies of the gene with the single nucleotide change, which could turn into a single amino acid change once it gets translated. Does that kind of make sense to everyone? Does anyone have questions about that? All right, so does anybody know how PCR can be used to test for COVID-19? Yeah. Yeah, so we would start by using primers that are specific for a gene in SARS-CoV-2. We would take a sample from someone who wants to be tested. And then what type of PCR would we use? Would we use standard traditional PCR? Would we use one of the other types of PCR? Yeah. Exactly, yeah. So SARS-CoV-2 has an RNA genome. So if we want to do PCR on SARS-CoV-2, we need to first use reverse transcriptase to turn the RNA back into DNA, which we can then copy using the DNA polymerase that we would use for PCR. What is the other special thing we need to do besides using reverse transcriptase? Yeah. Exactly, yes, a quantitative reverse transcriptase PCR, or sometimes it's called RT-PCR for real time. And so for this one, you use a probe that has a fluorescence, essentially. And so every time you make a copy of your gene, the probe gets a little bit more bright. And then once you make a certain number of copies of your gene, the fluorescence goes over a threshold for measurement, and then you can consider that sample positive. So you would, yeah. Okay. No, so I was just going to say, so you need primers, and you also need a probe that is specific for the gene that you're looking for. Yes. Uh-huh. Yeah. So yeah, so if you swab a person's mouth, right? Yeah, the cells in their mouth, the their DNA or their genome is made of DNA, but we're not looking for the person's DNA. We're looking for the virus's genome because we want to see if they have COVID-19. So the virus SARS-CoV-2, you're never going to see DNA for it in someone's body. You're only going to see RNA because its genome is made of RNA. So when the virus replicates, it makes more RNA into more RNA, and then it will make your body will help make that RNA into protein. But there's never going to be virus DNA for SARS-CoV-2. Does that make sense? Okay. Um, does anybody know? Yeah. Did you have a question first? Yeah. Right. So, okay. So if you have a cell that's infected with a virus, you'll have some DNA that's part of the person's genome. That just tells you about the person, what genes are in the person. And then if that cell is infected with virus, it'll also have RNA from the virus. This test that we're doing in this question is we just want to see if this person is infected with virus. It doesn't matter what the person's genome has in it, right? Because the person's genome is always going to be their genome. It doesn't tell you if they're sick or not. You only know if they're infected if they have viral RNA in the cell also. Does that kind of make sense? Like you don't need to make the person's DNA into RNA. Okay. Yeah. So reverse transcriptase. Yeah. Simple name. Yeah. Any other questions? Does anybody know? Oh yeah, go ahead. Uh huh. Yep. That's right. And you do that with reverse transcriptase, and that makes what's called cDNA or complementary DNA because it's complementary to the RNA. And then from there, your Taq polymerase can take over and start making copies of that DNA. Okay, cool. Yeah, it does not. Yeah, this particular virus doesn't. Some viruses do have DNA in them. There are DNA viruses and there are RNA viruses. So some viruses go through that sort of regular cycle that we've talked about where they have a DNA genome that gets made into RNA and that gets made into protein. Other viruses kind of skip the DNA step. Their genome is RNA. When they want to replicate, they just make RNA into more RNA. And when they want to make it into protein, they use our ribosomes to make that RNA into protein. They never have during their own life cycle, they would never make DNA of themselves. Yeah, so that viruses that have RNA genomes have a special type of RNA polymerase that can read off of RNA as a template and make more RNA. So it's what you call an RNA-dependent RNA polymerase because it's an RNA polymerase, right? It makes RNA and it depends on RNA as its template. And so most or all RNA viruses will have a gene that codes for this special polymerase because like when they infect us, we don't have that type of RNA polymerase because we never need to copy RNA into RNA. They need to bring that gene with them to tell us to make that protein inside of our cells. And then once that protein is made, it can then copy the virus's RNA. Does that make sense? Okay. Any other questions about that? Okay. Does anybody know what genes we would use from SARS-CoV-2 when we're doing a diagnostic test? Yeah. So yeah, often we'll use the spike protein. Um, what would this is sort of like not something I've necessarily told you specifically, but do you know why it could potentially be? Yeah, I guess maybe not. Never mind. So Yeah, I was gonna say, like, do you know like why you'd potentially have an issue looking for spike protein? But that's not really a problem with PCR tests. It's really more a problem with antigen tests. What I was thinking is that um, the vaccines that we make against SARS-CoV-2 are vaccines that have the spike protein in them. So if you're looking for spike protein in someone's body, it might be actually from the vaccine and not from the virus itself. But if you're looking at specifically RNA, the vaccines contain so little RNA that that wouldn't come up if you're doing a normal PCR test. So yeah, spike is a great gene to use for the PCR test. The other helpful gene to use is the nucleocapsid protein, because there is a lot of that protein being made. There will be a lot of RNA around for the nucleocapsid protein also. Any more questions about qPCR or diagnostic tests? Okay. So that brings us to DNA sequencing. So there are going to be 4 of these questions on DNA sequencing. I'll give everyone maybe 5 or so minutes to look through these, and then we'll come together and discuss them. All right, so does anybody want to go for the first question? So for DNA sequencing, for that when we're using the chain terminator method, what additional reagent do we use beyond the ones used for regular PCR? Yeah. Yes, DDNTPs. And these are dideoxynucleoside triphosphates. It's a mouthful. Do you remember the difference structurally between DDNTPs and regular DNTPs? Yeah, exactly right. So let's look at a picture of that so it's clear for everyone. So at the top we have a regular DNTP, it's a deoxynucleoside triphosphate. This is what we normally use during PCR to help extend our DNA chain when we're making the copy, right? We have 4 of them for all 4 of the different bases that you'd find in DNA. And they're deoxy on the 2 position, but they have this 3' hydroxyl group, which is important because that's what allows the DNA polymerase to keep extending the DNA backbone, to extend the chain of DNA. So for chain terminator sequencing, we have dideoxy. So these not only have this first deoxy, they have a second deoxy. So that's what the di is for. There's two deoxys. And the second deoxy, like you said, is at the 3 position. So if there's just an H here, there's no hydroxyl group. The DNA polymerase can't extend the DNA chain. The DNA stops here at this base. So when you have this, you'll end up, if you have mostly DNTPs and some DDNTPs, you'll end up, after you go through your reaction, you'll end up with a whole bunch of segments that are all different lengths, because some of them, depending on when each DDNTP got incorporated, the chain will stop there. So you will end up ideally with a chain of every single length, starting with like where you started from your primer, and then adding on 1 more each time until you get to the whole length of your template. And that's kind of the idea behind the chain terminator sequencing or Sanger sequencing. So to answer question number 2, how do we visualize the fragments that we end up getting from our chain terminator sequencing? Yeah. Yeah, so you can use fluorescent tag. You can also just use a colored, like just a regular dye, either one. And they're on each DDNTP is a different color. So that's kind of what we see here in this picture again. So our C's are blue, our A's are green, our T's are red, our G's are yellow. So now each different fragment is a different length, and it, depending on where the last nucleotide that you added in, it's a different color. So how would you then separate out all these different fragments to see the differences between them? So like right now, you just ran this big PCR reaction. You have, a tube full of fragments. They're all different colors. What do you do with them to see all the different ones and how long each one is? Yeah. Yeah, exactly. So you run a gel. You use gel electrophoresis to sort them by size. So that's one way you can do it. You can run the whole reaction in one big tube and have 4 different colors and then sort by size on a gel. The other way you can do it, so that's one way. The second way is to run 4 different reactions and each one of the different reactions has a different DDNTP in it. So you'll have one tube that's just, DD-TTP, one tube that's just DD-CTP, one tube that's just DD-ATP, one tube that's just DD-GTP. That's all 4 of them. And then after you run each of those 4 reactions, you would still run them on a gel. That way you can sort them by size. And you'd put each different DDNTP in a different lane on your gel. So you know that, everything in lane 1 has a T incorporated. Everything in lane 2 has, a C incorporated. And then you can use those to sort by size, and knowing which tube had which DDNTP in it, you can then work out what the sequence is. Does anyone have questions about that? Does that make sense for everyone? Yeah. Mm-hmm. So for this reaction, like most of the time, you use both, right? So this is sort of the list of everything you're using, right? Just like regular PCR, you have a template, a primer, your polymerase, your regular DNTPs. But you also will include small amounts of DDNTPs in addition. So you want at the end of the day to get chains that are of varying lengths. So for example, this chain on the bottom, you started out with your primer, GG, and then you added on just one C and it ended because you added the DDNTP. But you also want to be able to make ones that are longer, and you want each one to be longer by 1 nucleotide. So the ones that you incorporate, these first, whatever that is, 9 or 10 of them, are going to be regular DNTPs, but then the last one is going to be a DDNTP. So exactly, yeah. That's right, yeah. Yeah, exactly, because we're using the DDNTPs to tell us the length and also what nucleotide is getting incorporated at each position. Okay, yeah. So yeah, you put them all in together, and then just sort of by, I guess, randomness or having the correct ratio of DNTPs to DDNTPs, you should end up with, if you let it go long enough and you make enough copies, eventually you should arrive at having at least one, if not more, chains of every single possible length. And you need to make a bunch of each chain at each possible length, because when you run it on a gel, to actually be able to see the band, you need like a bunch of copies of it. Yeah, 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 which is sort of where the next couple of questions come in. So thank you for leading into that. But I guess before we go into that, does anyone have questions just about the first 2 parts of the question? Yeah. No, you're good. The 4 separate reactions one. Yeah, I think we actually have an example of that in the next part of this question. But I don't know if I should. I think if we wait until, do you mind if I explain that when we do that part of the question? Because I think it'll make more sense, like if you see it as we do it. Okay. Yeah. So it's the dye is just attached to each different DDNTP. So like on this picture, all of the DDATPs are green. So the color isn't dependent on the length, it's dependent on which nucleotide you put there. And then the lengths come along because it's copying a template, right? So they know where to go because they're complementary to the template, and then they're a color because each individual ddNTP is a different color. Yeah. Yeah. I'm sorry, what? Okay. I see. That's just unlikely. So I think that's also just a question of like your ratio of like regular DNTPs to your special ddNTP. So you want to have, enough there that you can make copies of all your different lengths, but not. But yeah, that's also a question that we'll get into based on parts 3 and 4 of this question. So like if you have too much or too little, something will go wrong in your reaction. You want like the right ratio of DNTPs to ddNTPs. So I think you will answer your own question, I'm saying, by the end of this slide, if that helps. I don't want to like tell you now because that's like the exercise, right? So does anybody want to make a logical, or do you have a question or did you want to answer? Okay, yeah, so you want to go for number 3. So what would happen if too much ddNTP was there? Exactly, yes. That was exactly the scenario that you were just suggesting, right? If you have too much ddNTP, it would be more likely to get selected very early on when it's building the chain, and you wouldn't be able to build longer chains that have the ddNTP encoded later in the chain. Does that kind of make sense? Okay. So if you have too many ddNTPs, you would never see what those longer chains look like. So you'd never see the end of your sequence. You'd only get, maybe the first few nucleotides, and then you'd be stuck. So you have to repeat it with fewer ddNTPs. So does anybody want to know, or yeah, do you have a question about that? Or you want to answer the next one? Okay, yeah. Oh, so like if one of the chain lengths was missing, is that what you're asking? Then you would see like basically a gap in your gel, which will also make more sense when we go over the next part of the question because I have a little picture of the gel and what it would look like. So as you run each chain length along a gel, right, they separate by size. So your shortest chains will run to the bottom, and then each one as you build up by one will be a little bit higher up the gel. So like let's say you're missing one that's 32 nucleotides long, you wouldn't see a band at the 32 nucleotides location or whatever. Does that make sense? If there's not dye. Oh, so if you run the 4 separate reactions, like if you have one ddNTP in each of 4 separate tubes, you wouldn't need a dye because you already know for the length for each of those chains that terminates which ddNTP went there. You don't necessarily need a dye as long as you run 4 separate reactions. If you want to run everything in one tube, that's when you need dyes because you need to know the difference between each of the 4 bases. Does that make sense? Okay. Finally, everything clicks. Okay. If it still doesn't make sense after the next couple of slides, like ask again. But I think it will make sense as we go through this. So does anybody have an idea for what would happen if we have too little DDNTPs. Go ahead. Yeah.
So you just get a bunch of long sequences, and you'll be able to tell where it's starts, and so you have just a bunch of
Yeah, exactly. So you would have more long chains, and you would also have gaps in the sequence. So if there aren't enough DDNTPs, they won't be able to incorporate at every single potential chain length where they belong. You would end up putting regular DNTPs there and the chain would keep extending. So you would get more of the longer chains, not enough of the short chains. And you would, even with the longer chains, you could still have gaps where they don't necessarily get incorporated because there's just not enough of them to randomly fill in there. Yeah. 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 Yeah.
this isn't necessarily going to go well. Sometimes, like it's
Yeah, it's like I think it probably depends on the particular DNA sequence you have and like how many DNTPs to DDNTPs, that particular ratio. Some sequences are more AT heavy, so you might need more A's and T's. Some are more GC heavy, you might need more G's and C's. So yeah, like you say, it's that's helpful to know that in practice, like you have to sometimes maybe repeat the reaction with different ratios of your reactants to get the right balance to fill in your full sequence. Thank you. Yeah.
you have too much, it's like every chain is supposed to be shortened or not, or like little, is it like
Uh, I wouldn't say either one is worse than the other. I mean, you don't want either, right? You want the right amount, like a yeah, like Goldilocks, right? If you have too little or too much, you're not going to get all the answers you want. Because if you have too short sequences, you won't know what happens later on in your template. If you have too many long chains, you won't know what happens earlier on in your sequence. So neither one is good if you want to know your whole sequence, and neither one is better or worse. Yeah. Any other questions? All right. So let's talk about another example here. So consider a template that has the sequence from 5' to 3' G, G, A, C, T, A, which is after the primer. So if we carry out sequencing using the chain terminator method, and we're going to use the 4 separate reactions method. So we're going to set up the same sequencing reaction in 4 separate tubes. Each one has a different DDNTP in it. Then each reaction is going to be run on an agarose gel in a separate lane. What would that gel look like? So I will go on to the next slide that has sort of a layout of everything from this question. I'll give everyone a few minutes to think about it, draw out their potential gel, and then we'll figure it out. All right, so yeah, so let's get started on this one. So when we started off with our template, right, we wrote it off in the 5' to 3' direction. That's the sort of standard way that we write out DNA or RNA. But when we want to write out a complementary sequence, it's sometimes helpful to flip the template around. So we have 3' to 5'. And that's part of why it's always important to label your ends, right? You always want to make sure you know which end is 5', which end is 3', in case somebody switches it around on you. So always pay attention to that. But if we write our template 3' to 5', that makes it easier to fill in our complementary sequence from 5' to 3', because that's the direction that it's going to get synthesized by DNA polymerase, right? DNA polymerase is going to make this complementary sequence going this way. So it's helpful to think about it that way. So can anybody tell me the first nucleotide that we would see in our complementary sequence, starting with the 5' end? Yeah, T. What's next? Oh, right, sorry. So let's take this one at a time. So yeah, so we know our first nucleotide is T. We know that that's the shortest possible chain, right? It's one nucleotide long. So if we had our DDNTP incorporate at this first position, at this T, it's gonna, when we run our gel is gonna run this way. Our smallest fragments are gonna end down at the bottom. So if we incorporated a DDNTP with a T at the very first position, it would end up at the very bottom of our gel in the lane where we ran the T's, because this reaction tube only had DDTTP in it. So anything that terminates in a T will be in this first lane. So and then any questions about that so far? Okay. So next, as you identified, we have an A. So where would the band for that A show up? You're raising your hand? Yeah. Yeah, it's a little bit higher and it's in the A lane, right? Because now it would come from our reaction tube that had DDATP in it, because it ends in A. So then our next longest chain would terminate in what letter? G, yeah. And so our G would then end up in, yeah, a little bit higher and in the lane with the G's. So I won't belabor this, but the next one is a T, so it's going to end up back in our T lane a little bit higher because it's a little bit longer. Our next one is going to be a C, so it'll end up in the last lane with the C's. It'll be a little bit higher. And then our last one is also a C, and it will end up just above there. Yeah. It's an interesting question. Yeah, I guess it's possible that you could end up with, for the full length band, if you happen to incorporate a regular DNTP there, it could show up in one of the other lanes. That might also be like a numbers game. Like if you hopefully would have more of them that ended up with the DDNTP in it. But yeah, that's an interesting point. I haven't thought about that if you run the 4 separate reactions, because it's not dye-based. Yeah. I will think about that and get back to you. Yeah. Yeah. Separate. Yeah, great question. So for a lot, for most people, if you run like a DNA gel in a lab, it's very, very difficult to tell the difference between chain lengths that are only one nucleotide different, right? These types of gels that are used for sequencing are really different types of gels. They're extremely long often, and that's what gives them the resolution to tell you the difference between different chain lengths that only differ by one nucleotide. So they're often very long and they're often very thin, like in these long tubes. They don't have to be in tubes, but they often are. And so yeah, if you should have good enough resolution on your gel that you would see two distinct bands there. You shouldn't see a smear. Yeah. Yeah. Right. So you shouldn't see, for every position, there should only be one correct incorporation of a nucleotide. So you should only see it showing up in one lane at that particular chain length. I mean, this reaction is being done by an enzyme, right? DNA polymerase is attaching them on. Every once in a while, DNA polymerase does make a mistake and incorporate the wrong nucleotide, but that's really, really rare. You wouldn't see enough copies of that mistake to actually be bright enough to be visible as a band on your gel. Any other questions? Does everybody now understand how to visualize what their gel would look like based on chain terminator reactions in 4 separate tubes? You may see this again. That is my hint to you. All right, so that being said, let's move on to the next part of this question. So we've been talking about the chain terminator sequencing, and now we're going to talk a little bit about the second generation sequencing or next gen sequencing that we discussed in the lecture. So I'll give everybody a couple minutes to think about these questions. So the first question is, what's the difference between the high throughput sequencing techniques from the traditional technique that we just discussed, the chain terminator, and why might it be useful to get a whole genome sequence for an organism? So I'll give everybody like 3 to 5 minutes for that. right, so can somebody tell me something that's either the same or different between the first gen and next gen sequencing techniques? Yeah. Mhm. Yeah, excellent. So first gen techniques are better for shorter sequences. Next gen techniques are better for longer sequences or whole genomes. Any other similarities or differences? Yeah. Exactly, yeah. So they both use PCR. So they're both going to use, a template, primers, polymerase. The ingredients are mostly the same. Any other similarities or differences? Yeah. Yeah, more often. You can, I guess, use fluorescent tags even for Sanger sequencing if you wanted. But um, yeah, I guess and you don't necessarily use fluorescence necessarily. I mean, you use like luciferase for pyrosequencing, right? Which doesn't necessarily, it's not really a fluorescent signal. It's light, but it's not fluorescent. Yeah. But it's a good thing to think about is differences in fluorescence. But it's not necessarily first gen versus next gen. So something we did on just now with our chain terminator sequencing is we ran a reaction and then we sorted it by size on a gel. Do we do that for next gen techniques? How do we see our results for next gen techniques? Is that the same or is that different? Yeah. Yeah. So for next gen, we don't need to sort. We see the how do I put this? So we can see our reactions as they go. We look at the color changes as they're happening compared to our chain terminator sequence where we did everything and then we had to run it on a gel to see what happened when or where. For next gen techniques, we're watching these changes happen like in real time. Like as the nucleotides get added on, we're seeing flashes of light or we're seeing different color fluorescence. The wrong shade of green. And so, sort of as part of that question, so if we just talked about for our first gen sequencing, we had to use those ddNTPs to make a whole bunch of different chains at different lengths. Do next gen sequencing also need to make a bunch of chains at different lengths, or do they do something different? So for next gen techniques, we're sort of like we were just saying, you watch them as they go, right? You don't need to make a chain of a certain length and then look what color it is. As you add on nucleotides to your chain, you will see whether or not it incorporates. So in pyrosequencing, you see if that nucleotide incorporated by a flash of light. For Illumina sequencing, you see which nucleotide incorporated based on the color that you see in that position on the surface that it's on. So you see the reaction as it goes. You don't make a whole bunch of different lengths and then sort them. You just you just watch them go. I just blanked out on why I was telling you that. It's been a long day. So for first gen, we need the DD NTPs to make different chain lengths, right? And in the next gen techniques, you don't, right? You just keep going and then you look at the changes as they go. You don't need to stop in the middle. So any questions about that or any other similarities or differences to add on to that? All right. So why is it useful to obtain the entire genome sequence for an organism? There's not like one right answer here. There's a number of reasons why you might. What is throw anything out there? Yeah, exactly. So you can look for how genetic changes affect an organism. So if you have organisms that are behaving differently or there's some kind of difference between them, you might want to sequence their genome to see is there a change in a gene or two or three that is somehow making them appear different. So that's what we call a phenotypic difference, right? A phenotype is a difference in an organism that is a result of a genetic change. Any other reasons why we might want to look at whole genomes of organisms? Everybody's tired today. Yeah. I'm sorry, speak up. Yeah, great example. So we can use genome sequences to compare different organisms and create a family tree, right? See how closely or distantly they're related to one another. So maybe like susceptibility to particular diseases. Or if we're looking for ancestry. So some diseases are genetic. So once whole genome sequencing became a useful technology, it was used to sequence, individuals who had a particular disease, and can then be helped to identify what gene might be causing that particular disease. Okay. Any other thoughts or questions here? Great. So let's talk about restriction enzymes. So first of all, what are restriction enzymes? Yeah. Yeah, exactly right. So they're enzymes that cut DNA at specific recognition sequences. Does anybody remember where these enzymes came from? Where do we find them originally? Yeah, bacteria. They're bacteria-derived enzymes. Does anybody, yeah, yeah. Yeah, so yeah, I guess that's not the only reason that restriction enzymes exist, but one of the reasons is that, so essentially bacteria, just like us, can get infected with viruses, for example. And so viruses might have differences in their genomes from bacteria. So the sort of bacterial immune system, part of it is recognizing certain sequences that are seen in maybe a virus that they've been infected with. And so, and if they recognize that sequence, they eventually developed, over time or whatever, the enzymes that would recognize specific sequences and cut them. Yeah, I mean, it's essentially a way to say, like, it's an immune system in the sense of, like, I see this thing, I recognize it, that it's bad, I want to get rid of it. So, yeah, and that's not the only thing restriction enzymes are used for. There's actually a whole bunch of different types of restriction enzymes that bacteria have. So some of them are used sort of for defense. Some of them are also used to control like the expression of bacterial genes that belong to the bacteria. So yeah, but does that make sense then? Okay. Yeah. So for those recognition sequences from the restriction enzymes, does anybody know what is special about the recognition sequences? Yeah. Yeah, exactly. They're palindromes. Or they're palindromic. And so that means the 5' to 3' sequence of the recognition sequence is the same on both strands of DNA. So if you had one that said GACT from 5' to 3', the or that's a really bad example because that's not a palindrome. Yeah, so it would be, oh, I'm so bad at this. You know what, we're going to look at some examples in a minute because my brain isn't working right now, but they read the same forward and backwards. It'll make sense in a few minutes. And restriction enzymes are a type of enzyme that's called an endonuclease because it cuts in the middle of a strand of DNA. So you might have heard of exonucleases. Exonucleases cut at the end of a DNA strand. Endonucleases cut anywhere that's not the end, so somewhere in the middle of a DNA strand. Okay. Yeah, sorry. Of this particular class of restriction enzymes. The ones that we have to be concerned with for this class, yes. Yeah. They're not just E. coli derived, so the name of each restriction enzyme tells you what bacteria it's derived from. So like EcoR1 is from E. coli, but you'll see there's a whole table of them that if it's not in these slides is in your book, and those are all from all different types of bacteria. And you don't need to memorize those sequences, like you'll be given a table. Any questions on this part? Okay, so I did not do this well. I'm just going to erase this so we can read it. Okay, so the recognition sequence for BAMH1 is GGATCC. Where in the sequence below will it cut? So, did you want to answer? Or, yeah. Yeah, like the between the fourth and the fifth on the top strand. Yeah. So that's, yeah, so here's the recognition sequence. I'll make this a different color. So the recognition sequence is this, right? So it's GGATCC. So if you read that 5' to 3' on the top strand, right, it's GGATCC. If you read it 5' to 3' on the bottom strand, it's also GGATCC. So that's what I meant by palindromic. And so for the, and so this recognition sequence has a vertical line written in it, right? So the vertical line tells you where the enzyme is actually going to cut the DNA. So it cuts in between the two Gs, and it does that on both of the strands. And so now you end up with this top part here, and then this bottom part cuts here. That makes sense for everyone. Does anybody have questions about restriction enzymes, cutting sites, or palindromes? Okay. So this cut was made by the restriction enzyme BAMH1. So did BAMH1 make blunt or sticky ends? Yeah. Sticky ends, yeah. And how do you know that? Yeah, exactly. So they're called sticky ends because one of the strands is longer than the other strand. So you'll have one strand that says CTAG on the top and GATC CTAG. So you have these four nucleotides kind of hanging out, and they really want to hydrogen bond with another DNA strand that they match with. So that's why they're called sticky, because they really want to stick to something. So I just drew out the two strands after they get separated, right? So each one has a sticky end that really wants to hydrogen bond. Yeah. Exactly. Yeah. So a blunt end would be if the each strand was the same length at the end, if it just cuts right down the middle of the two strands. Any questions about that? All right. So now we have our table of restriction enzymes. You will see this a lot. So the question is, what other restriction enzyme produces the same sticky ends as BAMH1? So I'll give everyone maybe 5 minutes, and then we will go over that. All right, so to start out, how do we know what the sticky ends are that the MH1 makes? Because we need to know what we're trying to match to, right? So yeah, so does anybody want to explain to me how you can use this table to figure out BAM-H1's sticky ends? Exactly, yeah. So you have the recognition sequence right here. Here's BAM-H1, here's the recognition sequence, and the little arrow tells you where it's going to cut. And it only gives you the top strand of the recognition sequence. You have to assume the bottom part is there. But yeah, that's exactly right. So you have G, G, A, T, C, C. You know, the complementary strand would be C, C, T, A, G, G. And we know that it's going to cut right in between those two G's. So what would our sticky ends look like? You can sort of pull the two strands apart, like in your imagination, right? So you have your 5' strand has just a G, your 3' strand has C, C, T, A, G, and they are sticky. For the other part of it, you have G, A, T, C, C, and then you have your G on there. Sorry, that's 5. So your G, A, T, and C on the top are sticky, right? They're looking for someone to stick to. So if you wanted to not have to draw all that out, right, you could just look at the table. So you know that in this recognition sequence, one nucleotide in is where the cut goes. So you know from the sort of the opposite strand, the cut would come in from one nucleotide in from there. So you sort of know that your sticky ends are going to be the central part, right? The sticky end says GATC from 5' to 3'. Does that make sense to everyone, how that kind of works? So is there another restriction enzyme on here that makes the same sticky ends, G, A, T, and C? Yeah. Yeah, exactly. Yeah, it looks like a 3. It's actually BGL2. It's really hard to tell with this font. That is not your fault. But yeah, so it has the same sticky ends, right? It has the central GATC, and we know that from this arrow, the cut happens right after this first nucleotide, and on the other strand, it happens right after the first nucleotide. That makes sense for everyone. Any questions? All right. So we have a couple minutes. So I think, yeah, so let's just talk about DNA gels for like a minute or two, and then we'll continue with this on Thursday because I don't want to fall behind. So here we have a DNA gel. So you will see a number of different types of gels in this class. Some will be DNA gels. We're going to talk a lot about protein gels in the next couple of chapters. One good way you can tell the difference is DNA gels are usually pictured this way with a dark background and bright or light colored bands. So that's how you know it's a DNA gel, and that's because of the way we often visualize the bands, which is one of the questions we're going to think about for this slide. But just keep that in mind. If you're ever not sure what kind of gel you're looking at, you have a dark background with bright bands. It's probably a DNA gel. Not always, but a lot of the time. So we've talked a little bit about this earlier today when we were talking about Sanger sequencing. By what property does gel electrophoresis primarily separate? Yeah, by size, exactly right. So size is the main way we look at them, right? We know that smaller or shorter length ones are going to be at the bottom of the gel. Longer chains are going to be near the top of the gel because they are running in this direction. And the other properties that we can sometimes separate by are charge and shape. In the case of DNA is mostly just uniformly negative charge because of the sugar and phosphate backbone. So all those phosphate groups are negatively charged. So you can always assume that DNA is negatively charged, so we don't have to worry about charge for this gel. So, I mean, that's how electrophoresis works, right? You have an anode on one side, and it will attract or other way around. You have the cathode that attracts negatively charged molecules, and they will get pulled that way. I always mix up anode and cathode. And the other thing that gels can separate by property-wise is shape. So for most of the time when we're looking at DNA, it's linear. It's just one long strand, so we don't generally worry about shape in DNA gels. But that is something to consider, mostly when you're working with plasmids is one common way you'll encounter this, because plasmids are circular. So depending and plasmids have this property where they like to do something called supercoiling. And when plasmids are supercoiled, they're basically all twisted up into like a really tight shape. And so they'll actually travel further down a gel. They'll behave as though they're a smaller molecule because they're so like bunched up together. If you cut that plasmid and made it linear, it would actually appear higher up on the gel. It would look longer, even though it's actually the same. So you sometimes have to take shape into consideration with DNA gels, but it's mostly size. And all right, so yes. It's the bottom is positively charged. So the positive charge is going to attract negatively charged molecules like DNA. So they get pulled towards the bottom. Yeah. And the top is negatively charged. It'll repel the DNA. So yeah, essentially. So things that are smaller or more tightly bunched up are able to travel through the gel more easily. So they will travel further in the same amount of time, given the same like voltage of charge or whatever. So they would travel further. So everything starts up here, right? You load your samples in the top of the gel, and then they travel this way. So like there's a positive charge down here, there's a negative charge up there. Your DNA is running away from the negative charge towards the positive charge. And we are a couple minutes over, so I'll let you guys go. But so we'll definitely finish this by Thursday. Your quiz for chapter 3 is due next Monday. It is available starting today, but I'd recommend waiting until we finish going over the chapter in case anything comes up. And so next time we'll finish chapter 3 and hopefully get a start on chapter 4. And if you have any questions, I have office hours right after this.